So, good morning everyone. I'm VAILLANT BEUCHOT, postdoctoral researcher in Véronique Packy's team from IRCAN in Nice. I will present to you the results of our study entitled The Nifuroxazide, NFX, Rescues the Deleterious Effects Associated with Micos Instability in Disease Models. Through these 4 points, we will see them in details during the presentation. So first of all, 10 years ago, my team was following family with patients presenting frontotemporal dementia amyotrophic lateral sclerosis disease. This disease was caused by a particular mutation, the S59L, on the gene coding for the protein coilcoid helix domain 10, D10, an important protein for mitochondrial physiology. In fact, we discovered that this mutation induces a strong alteration of mitochondrial cristae in patient fibroblasts, and also in some mitochondria a total disappear of cristae. Moreover, we demonstrated that D10 is essentially located at cristae junction with immunoglobulin. So the discovery of this mutation was the first genetic evidence that mitochondria impairments lead to motor neuron disease. And 2 years after that, my team was focused on the study of micos for mitochondrial contact sites and organizing cristae organizing system. This is an important multi-subunit complex allowing the correct invagination of mitochondrial internal membrane into cristae, essential for mitochondrial respiration. And we published that micos complex is also altered in patient fibroblasts. So one of our main aim was to determine if we could potentially reverse this alteration. So to answer this question, our last PhD student, Baptiste Roper, developed a model of yeast without MIC60. MIC60 is a core component of micos complex, and without it, there is a strong alteration of cristae shape and of mitochondria internal structure, with a reduced growth rate of this yeast. And the only way to rescue this growth rate was to transfect the yeast with the MIC60 wild-type GFP, or to find a molecule able to restore the growth rates. So we performed high-throughput drug screening in a numerous molecule and found one molecule, interesting molecule, a nitrofuran, the Nifuroxazide, able to rescue, this is the red curve, the growth rate of the delta MIC60 yeast. It's an FDA-approved drug and now patented already used against bacterial infection. So we have now the molecule. It's time to test it in patient fibroblasts. So first, you have to know that in patient fibroblasts, they presented thinner mitochondrial network with fragmented objects, fragmented mitochondria. And NFX treatment rescued with a dose-dependent manner mitochondrial network length, alternate length network. Moreover, NFX is also able to rescue cristae disorganization in patient fibroblasts, as we can observe here in electron microscopy images. And we reproduce these results in motor neurons derived from patient iPSCs. So NFX rescues mitochondrial network length and cristae disorganization in patients and models. But does it have some partners? So to determine that, we perform an affinity chromatography with magnetic beads covalently linked to NFX molecule and put them with an homogenate of fibroblast proteins and selected one of the most enriched protein, KIF5B, an essential protein implicated in mitochondrial anterograde transport. So, thus KIF5B, the heavy chain of kinesin 1, is essential for NFX treatment curative effects. So, to answer this question, we transfected fibroblast with a small interference RNA against KIF5B and observed in patients, so in the resulting gray just here, that we totally lost the curative effects of NFX after siRNA KIF5B transfection. So NFX effects depend on the presence of KIF5B. But to go further, is NFX is able to modulate mitochondrial movement itself. So we used two models. The first one, to answer this question, the first one was the NSE34 differentiated cells, overexpressing empty vector flag or D10 wild type flag or the mutated D10 flag. Then the differentiation worked well despite the overexpression of D10 and mutated D10, as you can observe here. And a macro imagery developed by Frédéric Brault, the head of our microscopic platform, allowed me to determine the mitochondrial motil fraction, mitochondrial global movements. And there is a significant rise of mitochondrial movement is S59L differentiated cells treated with NFX. To go further these results, I have to present to you one last member of mitochondrial protein transport machinery, the syntaphyline in red just here. This is literally a brake, able to stop mitochondrial transport in both ways, anterograde and retrograde transports, especially in axons. And the protein levels of this protein are strongly reduced in every cell treated with NFX. So in conclusion of this panel, NFX is able to enhance mitochondrial movements by down-regulating syntaphyline protein levels. And we reproduced these experiments in motor neurons derived from APSC, patients' APSCs. But this time, we cultured these motor neurons in microfluidic devices kindly made by Derim Kilinch from Institut Pasteur de Lille. These devices allow the correct separation without labeling, of cell compartments. So the location around the cell bodies, the axons, and the terminations. And I reported that NFX induces an important significant increase of mitochondrial global movements in control fibroblasts and motor neurons, and also in patient motor neurons. And syntaphyline protein levels are strongly decreased in motor neurons treated with NFX. So in conclusion, NFX also can enhance mitochondrial axonal movement in this particular model by down-regulating syntaphyline protein levels. So to conclude, the S59L variant at the origin of an FTD-ALS pathology causes mitochondrial network fragmentation and crystalloid disorganization, and this alteration are rescued by NFX. NFX is also strongly linked to global movements because of the involvement of Ki5B in its rescue mechanism. Moreover, NFX has also an important impact on mitochondrial transport regulation itself by down-regulating the syntaphyline protein levels and by enhancing mitochondrial global movement. So you will be able to read these results in few weeks because they were accepted recently in Brain. And to go further in the future, in perspective, we will perform a preclinical study on a mice model my lab developed and described 5 years ago, an S59L mice model. We already described that this mice model presented mitochondrial myopathy, motor neurons and neuromuscular junction degeneration, and significant motor neuron death in lumbar spinal cord. And recently, we described that this mice displayed dementia-like behavior plus neuroinflammation. So this preclinical study will be made by the Clinique de la Souris in Strasbourg, and we will determine what are the potential curative effects in vivo. So now I want to thank my team member, of course, and every people who participated to this study, the organizers of the JR10, and also you for your attention. Thank you very much.
Thank you very much for this very nice presentation. Is there a question in the audience? very beautiful work. There is something I don't understand. It's probably my fault. The drugs is acting on the crista inside the mitochondria, if I understand well.
Yes.
How do you link the protein that are moving the mitochondria, connecting from the outside to the crista
Disorganization.
which is inside and which is the target of your drug?
Yes, you mean with K5B?
Exactly.
That's right.
Exactly.
Yes. Yeah. So we targeted K5B starting with the magnetic beads by selecting the but the direct link is K5B. So is just the heavy chain of kinesin 1, and kinesin 1 can transport, for example, other entities like ribosome subunits. So mitochondria needs adapters to be recruited directly in kinesin or dynein following the transport. We perform a proximity ligation assay to determine that in patient fibroblasts there is an augmentation of the distance between mitochondria, adapters, and kinesin. And this distance is rescued by NFX. So the direct link is just this protein, it's MyRO1 directly anchored to mitochondria.
Thanks for the talk. Could you tell us more about the potential targets of Nifuroxazide? Would you happen to have targeted STAT1 or STAT3? As I believe they have been described as targets of the drug. And STAT1 is also a modulator of programmed cell death, so just as a control.
So this time we only worked on K5B after the target to determine the potential target of the drug. We are recently targeting another protein already captured with the magnetic beads, the CAMK2. The delta. That was the most enriched protein. Was these two proteins, K5B and CAMK2. So we were focused on this protein for now. But it's an interesting idea.
Okay, thanks.
Great job. I just want to know if Nifuroxazide, you mentioned this Nifuroxazide here.
Sorry.
You see, it's a problem of location sometimes. And I just want to know if the Nifuroxazide you mentioned was a drug you were testing to improve the situation of the mitochondria. This is the case?
Yes, exactly.
So this is very interesting because I just checked because I didn't know it. It's an antibiotics.
Totally, yes.
So it makes sense because mitochondria is phylogenetically a bacteria.
Yes.
So it's clever, but there's a problem. The problem, this drug does not cross the blood-brain barrier.
Exactly. So, that's correct.
No, because I find great. This is a great job. So I said, well, now we need to move forward. So maybe you need to patent and to change and to make it cross the blood-brain barrier.
Exactly. So I just come back to the preclinical study. It will be in two parts. The first part is to made IP injection in mice to observe the alteration we could observe in particular in, for example, in heart or in other pathology. And then once we validated these results, we will develop a few weeks, months after that, protocol to try to cross the blood-brain barrier. But it's one of we have to answer to this question, yes. No.
Okay, beautiful work again.
Thanks.
Are you aiming just at the transport to the site of action for mitochondria, or also mobility within the final cell, the final synapse that it's looking at? Because critical for crystal structure is also ER mitochondria contact sites, which are highly mobile and which we're looking at.
Oh yeah. So if I understand well your question, we are just also for now. This is the beginning of a mitochondrial movement study, but we study global movements in every part of the cells. But that's really interesting because we know that effectively in synapses, near the synapses, there is an important need of mitochondria. And also the dynamic near the endoplasmic reticulum is also important, and it's one of our future aim.
Okay, well, I suggest we have a chat about that later on.